question can I ask, please?
Sorry, we're running out of time. Please.
I think okay.
Okay,
Yeah,
okay, just
go ahead,
last,
go
just
ahead, go
last.
ahead.
My question is, how does studying about astronomy help in our society? Like
Okay, that's an interesting question. I was expecting
studying
this
about
particular question from you.
exoplanets, stars, how does it help the society?
Okay, that's interesting. So everyone keeps asking, how did human or life came to this Earth, particular Earth? If we are looking at many of Earth, such Earth system, and we are looking at different phases of their evolution, we are able to understand how life or some life forming molecules are forming in those planets or in those systems. Understanding those, we will be able to understand our own evolution. How did probably how did human evolve? Not only that, whatever the fundamental laws people have given on the Earth, we are able to test those fundamental laws. Are they correct over the other planets, over the other system or not? So basically trying to constrain our own physics, chemistry, biology, everything. Yeah. Maybe I will take questions later. You can find me somewhere. We are running late. Yeah.
You can mail to outreach if you have any further
Yeah, if
questions.
you have
They
any
will be
questions,
replied. We
write an
promise
email
they
to
will be
outreach.
replied. Please be seated.
Thank you.
So, आप लोगों में से कितने लोगों को कीमोथेरेपी के बारे में पता है? Louder. You can shout. So हमारी नेक्स्ट टॉक के लिए आ रहे हैं श्रेयस जो कि जिनका डिपार्टमेंट है केमिकल साइंसेज और उनकी लैब का नाम है मेडिसिनल केमिस्ट्री एंड सेल बायोलॉजी. ही इज गोइंग टू टॉक अबाउट कीमोथेरेपी and chemotherapeutic drugs and how they work and how to improve their selectivity, because you know there are a lot of side effects of chemotherapy. So how to reduce those side effects and make them particularly work for the purpose they should work. So over to Shreyas.
Yep. Hello. Is it coming? I don't know. Am I audible? Yes. Okay. Okay. Yeah. So good afternoon to all of you. Thank you very much for the introduction. My name is Shreyas Vaidya. I am a graduate student in Medicinal Chemistry and Cell Biology Lab, DCS for Mumbai. And today I'm going to talk to you about chemotherapy, a double-edged sword against cancer. So to begin with, I have some people here. Do you know these people? Yes, very good. So this is Manohar Parrikar, former Union Minister. This is Arun Jaitley. His name, yeah. Arun Jaitley, former Union Minister. This is Rishi Kapoor, acclaimed actor. Professor Severus Snape. How many of you know Professor Severus Snape? Fantastic. Alan Rickman. And this is Irfan Khan, another legendary actor. Now, apart from the fact that these people were luminaries in their respective field, there is another thing that is common to them. Yes, all of them succumbed to cancer. Now, at this point, let me ask you one question. How many of you here are familiar or know of at least one person in your immediate vicinity who has been afflicted by this disease? I'll raise my hand. Okay. All right. So most of you know of at least one case in your immediate vicinity. So don't you feel that we should elevate cancer to a pandemic level? It is just that cancer cannot be transmitted from one person to another by mere coughing and sneezing is the reason why it has not been qualified as a pandemic like COVID, but it is there. Let's look at some statistics. So it is the second leading cause of death. Can anybody guess what is the first leading cause of death? Sorry? No? Cardiovascular disease. Fantastic. In 2020, we had 20 million new cancer cases and 10 million deaths. And this is how a normal cell divides, and this is a cancer cell division. Now, till this point, you might be under the impression that, oh, Shreyas is picturing or presenting a very grim picture. But it is not that. Have we lost the war against cancer? Absolutely not. So we again have a panel. I hope you people know these personalities. This is Sonali Bendre. She is an actress. Mahima Chaudhary, another actress. We cannot forget this guy, UV. Okay. ICC World 2020, the match against Stuart Broad, 6-6s, memorable. And then these two guys, these are acclaimed artists in the Marathi theater industry. मराठी रंगभूमीबद्दल ज्यांना माहिती असेल ते ओळखतील की हे आहेत अभिनेते अतुल परचुरे आणि हे आहेत अभिनेते शरद पोंक्षे. There is another thing, there is one thing that is common to all of them. And the thing that is common to all of them is all of them have fought the war against cancer and emerged victorious. So the bottom line is that most of the cancers are curable with proper diagnosis and treatment. So there is hope. I have a philosophy, whenever I start any assignment, whenever I tackle any problem, I ensure that I am clear about two things. First thing is, what is the problem that I am solving? And second thing is, I should have clarity about myself. So cancer is a multidisciplinary problem. People from different disciplines are working on this particular problem. So we have physicists, we have mathematicians, we have biologists, we have doctors. Now, doctors include both surgeons, oncologists, radiologists, and whatnot. And then here I am. Here we are, chemists. Okay. Now, I must mention here that the word chemist has got different connotations. Okay. My mom used to tell me, Shreyas, jaake chemist se dawai leke aao. So I am not that chemist. Okay. I am a student of chemistry. Rasayan Shastracha vidyarthi ahe mi. And what is my job? My job is construction and destruction of molecule. Molecule को जोड़ना है और molecule को तोड़ना है. इतना ही काम आता है हमें. So that is my work. So first thing is, थोड़ा clarity आ गया मैं कौन हूँ, थोड़ा clarity आ गया कि problem क्या है. Let's try to bring some more clarity. What are the two facets of the cancer problem? One is detection and diagnosis. Can we detect cancer? Can we diagnose cancer? Second vertical is treatment and palliative care. So in our lab, what we do is, we are medicinal chemists, as I told you in the previous slide, for working on the treatment and palliative vertical of cancer. Okay, great. So we have clarity. Now, let me tell you about the treatment modalities that are typically available in the clinic. So first is surgery, Shastrakriya. Yeah, so the first is surgery, Shastrakriya. Surgery, remove the tumor. Radiotherapy, you subject the patient to radiation. Typically, X-rays are used. Now, you will realize that surgery and radiotherapy are applicable provided, the contours of your tumor are well defined. If the contours of your tumor are not well defined, let's say you have leukemia, raktacha karkarog, okay? If you have leukemia, surgery, can you do? No. Can you apply radiation? No. Let's say you have metastatic cancer, cancer that has spread to multiple organs of the body. Can you do surgery? No. So it is in this context that this fellow, chemotherapy, comes into picture, where you treat the cancer by medication. And as you can see, chemotherapy can be applied to both localized as well as metastatic cancer. Okay. Now, let me tell you a bit about chemotherapy. Cytotoxic small molecule drugs. हा शब्द सायटोटॉक्सिक काय आहे? सायटोटॉक्सिक हे कॉम्बिनेशन आहे दोन ह्याचं. सायटोस म्हणजे पेशी, सेल्स. टॉक्सिक विष म्हणजे अशी रसायनं जे तुमच्या पेशींसाठी विषारी ठरू शकतात. ऐसे केमिकल्स जो आपके सेहत के लिए हानिकारक होते हैं. साइटोटॉक्सिक स्मॉल मॉलिक्यूल ड्रग्स. ओके, हियर आर सम स्ट्रक्चर्स. आई डोंट वांट यू टू गेट इंटिमिडेटेड बाय लुकिंग एट द स्ट्रक्चर्स. आप सिर्फ देखिए कि टैक्सोल नामक कोई रसायन है. ओके, क्लिनिक में जाएंगे अगर आप और कोई आपको अगल बगल में बोलते हुए दिखेगा कि वो पेशेंट एक्स हैज बीन एडमिनिस्टर्ड विथ 5 milligram per kg of Taxol, that time you should remember that, oh, somewhere in TIF or Mumbai you have seen Taxol looks like this and it's a chemotherapeutic drug. This is doxorubicin, etoposide, cisplatin. These three molecules come under the class of organic molecules, carbon, nitrogen, oxygen. How many of you are aware of organic chemistry, by the way? Fantastic. Very nice. Very nice. So these are organic molecules. Cisplatin is an inorganic molecule. Chemotherapy का महत्व क्या है? Clinical impact. 50% of cancer patients are treated with platinum-based drugs. I mentioned in this last slide that they are effective against both localized and metastatic cancer. They have broad anti-cancer activity spectrum, which means that the chemotherapeutic drug can act against a variety of cancers, so head cancer, neck cancer, gallbladder cancer, and whatnot. In the case of testicular cancer, particularly, cisplatin's cure rate is phenomenal. You see, greater than 90%. Then, very important, it is medicine for the common man. Now, I'll put this point in perspective. Cisplatin, the guy that I have shown here, can anybody hazard a guess about its cost? Kai guess? किती रुपयाचा असेल cisplatin? 100? 100? 500 to 1000. Very nice. 500 to 1000. इसका एक भाई है जिसका नाम है कैटसाइला. Now, Kadcyla belongs to a category of molecules that is called as antibody drug conjugates, ADC. And Kadcyla's cost is 1 lakh per vial. So you can imagine, cisplatin is 500 rupees per dose per vial, and Kadcyla is 1,15,000, around 1,15,000 per vial. So there is a huge difference. And so Kadcyla is something that cannot be afforded by common man. Okay, it is reserved for the elite class. But cisplatin is something that can be afforded. Okay. What I showed you in the previous slide was the upside of chemotherapy. Okay, what are the benefits of chemotherapy? But just as a coin has both head and tail, chemotherapy also has a flip side. So let's talk about the flip side. First thing is resistance, विरोध. Resistance is the major reason for failure in cancer chemotherapy. जैसे आप chemotherapy दे रहे होते हैं एक patient को, patient's cancer stops responding to that chemotherapy. Now this resistance could be either pre-existent, that is intrinsic resistance, cancer में पहले से ही resistance होता है, so that is why it does not respond to chemotherapy, or it acquires this resistance during the course of treatment. So this is called as acquired resistance. And here you can see cancer cell is resisting, okay, all your attempts to administer chemotherapy. One thing. Second thing is selectivity. What did I do? What did I mention in my definition of chemotherapy? पेशंटना दिलं जाणारं विष, डन. ते विष तुम्ही घातलं एकदा आतमध्ये की त्याच्यानंतर देर इज गोइंग टू बी इनडिस्क्रिमिनेट किलिंग. ओके, once your chemotherapeutic molecule enters inside your body, it doesn't know whether the cell that is there in front of it is a normal cell or a cancerous cell. It is going to apply the principle of equal justice. Okay, and so what happens is when this indiscriminate killing happens, you get side effects. So here I have represented some commonly observed side effects. This is cardiotoxicity. तुमच्या हृदयामध्ये साइड इफेक्ट होतात. न्यूरोटॉक्सिसिटी, डोक्याच्या ज्या पेशी आहेत त्यांच्यात साइड इफेक्ट होतात. नेफ्रोटॉक्सिसिटी म्हणजे किडनीमध्ये साइड इफेक्ट होतात. ॲनेमिया, युअर रेड ब्लड सेल्स. अँड ऑटोटॉक्सिसिटी म्हणजे नैसर्गिक श्रवण यंत्र, कान, तिथे प्रॉब्लेम होतो. ओके, सो दीज आर द ड्रॉबॅक्स. नाव let us keep these drawbacks in our mind. Let us keep them in focus and ask ourselves, ideal chemotherapeutic काय असू शकतं? What is the notion of an ideal chemotherapeutic? So here again I have shown two pictures. One idea that we have in which we have is multi-targeting. So can you recognize this photograph? Oh, fantastic. थोडासा geopolitics, okay? Israel and Hamas. Okay, some war is going on. Okay, we'll not comment on that war, but let's see what is this. So what is this? This is Hamas that is firing multiple rockets towards Israel. I hope you are aware of this fact that Hamas fired multiple rockets towards Israel. Now Israel has a very powerful defense system that is called as the Iron Dome. Okay, now when I had a look at this war, the first question that came to my mind is, how is it that Hamas was able to overpower and overwhelm Israel's Iron Dome system? And what Hamas did was it fired multiple rockets. I said, okay, this is very nice. What we'll do is, this is an operational strategy, a war strategy. Let's lift up this war strategy and apply it in the cancer context. Because cancer cells also have an Iron Dome. So we'll keep this in mind. Second photograph here. Can you recognize this? Okay, this is Arjuna. Okay, many years back, Guru Dronacharya was teaching a course, Archery 101. Okay, so when he was teaching that course, Archery 101, what he did is he called all the Kauravas and Pandavas, and then he told them that, look, there is a parrot, you have a target, and now you describe. Before you shoot the arrow, please give the description. And then there were students who were like, I can see the mountains, I can see my Guru, I can see this thing, that thing. It is only Arjuna who told that, look, I only see the parrot's eye. So yes, I said, this is again fantastic. If you want to have a selective chemotherapeutic, the moment it enters inside your body, it should tell you that I see only the cancer cell, not anything else. So let's take this war strategy. Let's take this attribute, and then the notion of an ideal chemotherapeutic is molecular Arjuna that can execute a blitzkrieg on cancer cells. Molecular Arjuna, you understand? It should have selectivity of this nature. Blitzkrieg, how many of you are aware of blitzkrieg? German war strategy. Okay, so this is संकल्पना, हा विचार आहे. हा विचार आपल्या डोक्यात आहे. आता आपण बघूया की ह्याला रासायनिक रूपांतरण, chemical transformation, an idea, how do you construct or how do you convert it into a chemical identity? And then the third part of our research is, once you have the chemical structure with you, you test it under biological conditions. So just to give you a broad overview of what we do in our lab, medicinal chemistry lab, we work on these 4 areas, multi-targeted anti-cancer agents, and this is the blitzkrieg photograph. Okay, these are the molecules that go and do this blitzkrieg attack on the cancer cells. This is natural product chemistry. I work on natural product chemistry. This is bleomycin, and this is supposed to be our molecular Arjuna. Then what we do is, you take multi-targeted anti-cancer agents, you take these cancer-targeting vectors, you combine both of them. And so the third area of our lab is working on targeted delivery to cancer. And once you have the chemical structures with you, you do in vitro studies. In vitro म्हणजे cancer cells मध्ये केलेला study. In vivo म्हणजे animal model मध्ये केलेला study. What is the research methodology that we employ in the lab? First is design, synthesis. This is where the chemistry aspect comes into picture. You can see a beautiful flask here with chemical structures. Then once you have the molecules, we do screening. We test the molecules in vitro. From the initial screening, we get a promising compound that is called as the hit compound. And once you get the hit compound, you administer it to mice in vivo study. Okay, and then this cycle continues. And that is how we refine the chemical structures that we get. So let me give you 2 examples from our lab. First example is magic bullet to magic shotgun. What is the concept? Magic bullet. एक बुलेट घेतली, मारली, एका टार्गेटला हिट होणार. शॉटगन म्हणजे काय? Instead of indulging in this one bullet, one target, you bring a shotgun that can fire multiple bullets. Okay. Now this is abstract thought process. How do we realize this chemically? Cisplatin, well-known drug. This goes and binds to DNA and causes cancer cell death. There is another guy who is called as ferrocin. Ferrocin, what it does is, it catalyzes this reaction called as Fenton reaction. And this is the process by which this guy also goes and kills cancer cells. So what we thought is, take DNA binding cisplatin, take Fenton catalyst ferrocin, merge them together, and you get this particular molecule. So you see, this has platinum, this has ferrocin. This particular process in scientific medicinal chemistry parlance is called as polypharmacology or pharmacophore merging concept. This hybrid agent, okay, is supposed to be a lipophilic cation. Lipophilic cation is a technical term, so let's not worry too much about it. Now, what is it that we expect from this hybrid agent? Once you administer this hybrid agent to cell, it should go and attack 2 places. Mitochondria, which is supposed to be the target of this ferrocin, and since we have platinum, so it should go and attack the nucleus. So this is multi-targeting. Okay. Now, in vitro. Okay, so we have a hypothesis. We conceived a chemical structure. We synthesized the chemical structure. Now comes the acid test. Is your hypothesis really working? So in vitro potency. Simple question. If you don't want to get bogged down by the technical description, technical title, cell में क्या होता है? That's the question that we are asking. Okay. Now, this in vitro study has 3 phases. This is technically called as cell seeding. This is called as compound incubation, and this is dose response analysis. But we'll think about it in a different way. Cells है, पहले हम क्या करते हैं? Cells को लेते हैं, कैंसर सेल्स हैं ये, इनको अच्छे से खाना पीना देते, ओके, इनको अलीशान रूम में रखते, एकदम हैप्पी, ओके, बहुत अच्छा खाना पीना अलीशान रूम में रखते. टेक्निकली व्हाट आई एम सेइंग इज दैट you give them sufficient nutrients and you incubate or you put the cells inside an incubator, controlled atmosphere. तो इनको अच्छा सा खाना देते हैं, उसके बाद एज आई सेड अलीशान घर में रखते हैं, तो सेल्स बहुत खुश होते हैं. नाउ एट द राइट टाइम हम सेल्स के ऊपर सर्जिकल स्ट्राइक करते हैं. सर्जिकल स्ट्राइक मतलब क्या? वी ट्रीट द सेल्स विथ आवर टॉक्सिक मॉलिक्यूल, एंड once you do the surgical strike on cells, this is how it looks like. So this is pre-surgical strike photograph, this is post-surgical strike photograph. अभी ठीक है, आपने सेल्स को टॉक्सिक कंपाउंड के साथ इंक्यूबेट किया, आपने सेल्स के ऊपर सर्जिकल स्ट्राइक किया, बट आपका सर्जिकल स्ट्राइक कितना इफेक्टिव था, ये कैसे पता करेंगे? सो हमारे लैब में हम उसको डोज रिस्पॉन्स कर्व देख के पता करते हैं. सो the effectiveness of your surgical strike is determined by the dose response curve. Dose response curve से आपको एक वैल्यू आता है जिसको हम IC50 कहते हैं. तो the point which I want to make here is, the lower is the IC50 value, the more effective is your surgical strike. Simple. जितनी कम IC50 वैल्यू होगी उतना आपका मॉलिक्यूल ज्यादा टॉक्सिक है कैंसर सेल्स के लिए. Okay. These are the 2 molecules under consideration. This is the hybrid that I talked about previously, and this is oxaliplatin, control molecule that is available clinically. Now I want you to have a look at these values. Okay, we have tested this in A2780. अभी ये A2780 क्या है? Human ovarian cancer cell line. Okay, and you look at these values, near about same. You look at these 2 values. In A2780, this is 1.35, 0.72. Almost equally potent. But what I want you to focus on is this value in A2780 cis. So A2780 cis जो है वो resistant cancer cell line है. And you remember that the whole objective of constructing or developing this molecule was to overcome resistance. So if your molecule is able to exert its activity in resistant cancer cell line, that's what you want. So now you see, oxaliplatin in a resistant cell line says नहीं भाई, मुझे resistance आ रहा है तो I will not act. So its IC50 value increases, but this person PTCF32 is able to exert its activity, and this parameter is called as resistance factor. So the lower is the resistance factor, the more effective is your molecule in both normal and resistant cell lines. So ideally you want your molecule to have a resistance factor of 1. So if the resistance factor is 1, means that molecule जब आपके कैंसर सेल को देखता है और रेजिस्टेंट वर्जन को देखता है, it will say I can hit both of them with equal potency, no problem. That is the ideal yardstick. Okay. So ये तो सेल में हो गया. So सेल में हमें दिखा कि whatever hypothesis we had in our mind seems to be working, which is fine. But is the cellular picture the reality? No. So then what do we do? In vivo picture. In vivo picture means we ask this question, mice में क्या होता है? Elevate the study to the higher organism. So again, the molecules under consideration are this bimetallic hybrid and oxaliplatin, and then these are technical details. हम चूहा लेते हैं, फिर चूहे में ट्यूमर ग्रो करते हैं, ट्यूमर ग्रो होने के बाद चूहे को हम इंजेक्शन देते हैं. These are technical details. What I want you to look at are these plots. First thing is efficacy. Efficacy refers to the ability of a compound to produce the intended result. अभी यहाँ पे intended result क्या है? You want the tumor to shrink. That's it. So on the y-axis you have RTV, that is relative tumor volume. So you see, ये RTV का वैल्यू जितना ज्यादा होगा उतना उसका मतलब है उतना बड़ा है आपका ट्यूमर. Okay, and if the RTB value decreases, it means that the tumor size is shrinking. So look at the vehicle control. Vehicle control मतलब क्या? हमने चूहे को कुछ नहीं दिया है. Okay, there is no cytotoxic or chemotherapeutic molecule that has been administered to the mice. So उसमें तो ट्यूमर बढ़ते ही जाएगा. Look at 2, blue line and oxaliplatin, that is clinically available molecule. So you can see that the blue line is below the red line. During the entire course of treatment, there is this dip here, and I'll come to this dip a little later. But if you look at in general up to, let's say, day 22, the blue line is below the red line. So it means that the molecule is able to exert its in vivo activity, its activity under in vivo condition as well. And these are the actual tumor photographs. Vehicle treatment में tumor आपका ऐसे दिखता था, and this is the photograph in the compound treatment, compound treated group. Now, if your molecule is causing side effects, okay, the first visible indication will be drastic variations in the body weight. So body weight में अगर change आएगा, so that will be the first indication that your compound is causing some side effects. So, sorry. So I want you to have a look here. Body weight was monitored. If you look at the vehicle control and the compound treated group, body weight did not change significantly. But you look at oxaliplatin, there is a decrease. So चूहों को तकलीफ हो रहा है, चूहों को side effects हो रहा है. But in the compound, there are no side effects. And then the third parameter is survival. This plot is called as Kaplan-Meier survival curve. What you have to notice here, or what is of importance, is if you see a dip in the Kaplan-Meier survival curve, it means death. Okay, so you see oxaliplatin, there are dips in the Kaplan-Meier survival curve. So it means that the mice are dying over the course of treatment. But if you look at the compound treated group and the vehicle control group, there are no deaths. So overall, what have we observed? We have observed, or what we have found out, is that this bimetallic hybrid is able to exert its activity under in vivo condition. We saw the in vivo data for the normal cancer cell and the resistant version. Then we translated this molecule to in vivo studies, and under in vivo studies, we saw that it is able to perform better than oxaliplatin. There are no visible side effects, and there are no deaths during the course of treatment. So we were very happy. Yes. Oh, I have only 5 more minutes. My God. Okay. Now it's time to accelerate. Okay. So first generation I talked about. Second generation. While designing the second generation hybrids, we utilize this particular principle. Organic chemistry में बहुत बढ़िया principle है. Paula Bruce आप refer कीजिए. Reactivity is inversely proportional to stability. ठीक है, free radical hallucination का chapter है, उसमें you will get it. And if you think about this principle, this is not just restricted for organic chemistry, this is also applicable to life. जो जितना reactive है, उतना कम stable है. So what we did is, we utilized this principle in designing the next generation of molecules. So what we did is we toned down the reactivity of the molecule. हमने बोला भाई शांत हो जा थोड़ा सा, कम reactive हो जा. So we installed a CH3 group here instead of CF3. We toned down the reactivity of the molecule. We improved the plasma stability. Here, by stability, I mean plasma stability, because administration के बाद आपका molecule आपके plasma में घूमने वाला है. So it should not, have indiscriminate reactions. It should not show indiscriminate reactions in the plasma. So we improved upon the stability, and now you see here, the relative tumor volume decreased further. So hypothesis is working. So, and you see these are the actual photographs of the tumor. So tumor shrinkage bhi hua, and at the same time, you can see that these tumors appear to be whitish. Can you see that? These are reddish, these are whitish. What does this mean? This means that in addition to enhancement of tumor shrinkage, this molecule was also able to exert what is called as anti-angiogenesis. मतलब ट्यूमर को जाने वाली ब्लड वेसल्स जो है उनका ग्रोथ भी इसने कम कर दिया. This is fine, fantastic. In our lab, we have been working on molecules beyond platinum. These are some examples. So we have iridium-based molecules, platinum, ruthenium, rhenium. And I will tell you one thing. इसमें से हर metal की अपनी एक दास्तान है. Okay, and because of paucity of time, due to paucity of time, I will not be able to delve into the details, but each metal has its own story. And that is the beauty of chemistry. I'll quickly give you another example. This is an area in which I am working. Carbohydrates as sweet trap for cancer. We all know that cancer cells need food in order to sustain rapid proliferation and growth. अभी cancer cells का food क्या है? You can have carbohydrates, vitamins, amino acids, and so on. Out of all these constituents, we are interested in carbohydrates. So हमारा strategy क्या है? We'll try to fool cancer cells by making them gobble up sugar that is laden with chemotherapeutics. And this is the outline. So हम chemotherapeutic लेंगे, which has all these negative attributes. We'll take carbohydrates or sugars, chemotherapeutic carbohydrates, we'll conjugate them to get what is called as a glycoconjugate. And then you can see that there are these arrows, these things that are upregulated on cancer cells as compared to normal cells. So इनको आप दरवाजे की तरह मान लीजिए. Cancer cell के ऊपर ज़्यादा दरवाजे हैं. So if there are more doors on the cancer cell, it means that more of these carbohydrate cytotoxic molecule conjugates can go in. So cancer cells का भूख जो है, we'll use the hunger of the cancer cells as a differentiator to ensure that more number of your glycoconjugates enter into your cancer cells as compared to normal cells. So carbohydrate में काम करने का reasons summarize कर देता हूँ. First thing is, if you work on glycoconjugation, what you do is you improve the water solubility and biocompatibility. Carbohydrates are supposed to be more, or they are more soluble as compared to other molecules. So water solubility you improve, biocompatibility you improve. Second thing is that you bring in selectivity because the transporters that recognize carbohydrates are overexpressed in cancer cells as compared to normal cells. Third advantage, ये जो transporters है, which are overexpressed on cancer cells, you will see that they are overexpressed on a variety of cancer cells. So the advantage of working on glycoconjugation strategy is that whatever therapeutic construct you make will be applicable for a wide variety of cancers. So it will not be a cancer-specific chemotherapeutic. You can use it for a variety of cancers. And fourth thing is Shark Tank India. How many of you have watched that series? Fantastic. So in that series, there is a judge, Namita Thapar. So whenever Namita Thapar doesn't want to invest in any venture, she has this famous line, ये मेरा area of expertise नहीं है, इसलिए मैं out हूँ. But I tweak that statement a little bit and I say that ये मेरा area of expertise है, इसलिए मैं in हूँ because I'm a carbohydrate chemist. So it is because of these four factors that our lab is interested in working on glycoconjugates. And now you will ask me that, Shreyas, okay, this sounds very good, but कुछ clinical precedent वगैरह है क्या? I said, yes. Look at this molecule. This is called as 18-FDG. And this is clinically used for detecting cancers. Okay? PET scanning. PET scanning में 18-FDG use होता है. 18-FDG does the same thing. It exploits these transporters and goes and sits inside your tumor. So then the tumor is lit up. So your PET scan can, in your PET scan, you can see that, oh, this is the location of tumor. So this is in diagnostic purposes. Then there is another molecule called as glufosinamide. This has reached phase 3 clinical trials for pancreatic cancer. And those of you, I'm indeed very glad that most of the people in the audience are aware of organic chemistry. So this is a visual treat for you. Okay, structures. Just as equations and numbers represent art, beauty, and reality for mathematicians and physicists, chemists के लिए structures represent art, beauty, and reality. So these are some of the molecules that we have synthesized in the lab. There are many more in the pipeline. And when I was making this slide, nowadays you have this on the social media, there is this POV concept, some point of view concept, something like that. So when I was making this slide, I had a new POV. Okay, and what was this POV? So I asked myself, Shreyas, क्या बनाया है तुमने? Carbohydrate, cytotoxic molecule. अच्छा, कार्बोहायड्रेट सायटोटॉक्सिक मॉलिक्यूल. मराठीमध्ये भाषांतर करायचं. साखर आणि विष, विषारी साखर. ऐकायला छान वाटतं एकदम, विषारी साखर. चलो हिंदी में ट्रांसलेट करते. हिंदी में शक्कर, जहरीली शक्कर. अरे बाप रे, जहरीली शक्कर. बट देन आई एम अ बॉलीवुड बफ. ओके, जहरीली शक्कर, जहरीली खीर. समथिंग केम टू माय माइंड एंड देन आई वाज लाइक माय गॉड, ये तो रिपोर्टेड है. I hope that all of you are aware of this famous scene, Suryavansham movie, towards the end, when Thakur Bhanu Pratap takes this kheer, he has this reaction. Okay. And then the new POV that I had was, what am I doing in the lab? My job is to make zehirili kheer. Okay. That's precisely what I'm doing. I'm making zehirili kheer. And I have a cell biology colleague in my lab. What we do is, I make zehirili kheer, my cell biology colleague administers that zehirili kheer, and then we ask or we check if the glycoconjugates or the zehirili kheer that we make can elicit this kind of a response in cancer cells. That is what we are doing. Okay. Towards the end, I'll just tell you a little bit, touch upon some advanced treatment modalities, immunotherapy. Okay. I am sure that most of you will agree with me that cancer is an internal security problem. Okay, आपके ही cells है जिन्होंने आपके खिलाफ विद्रोह किया है. So if you have an internal security problem, it is best dealt by internal security forces. हिंदी मूवी है एक, आखिर क्यों. ठीक है, स्मिता पाटिल एंड राजेश खन्ना लीड पेयर है. तो उसमें एक बहुत भारी सॉन्ग है, दुश्मन ना करे दोस्त ने वो काम किया है, उम्र भर का गम हमें इनाम दिया है. तो यहाँ पे आपका दोस्त आपके ही सेल्स हैं, जब कैंसरस होते तो आप उनके लिए ये कविता समर्पित कर सकते हैं. इम्यूनोथेरेपी, आपका ही इम्यून सिस्टम आपकी मदद करेगा. एंड दिस इज अ स्कीमेटिक ऑफ CAR T cell therapy. Okay, I am not going into the details, but I will tell you that Indian scientists have actually made significant progress in CAR T therapy, and recently a group from the Indian Institute of Technology, Bombay, has been in news. Okay, you can go and check it out. Second is photodynamic therapy. Photodynamic therapy, as the name suggests, photo means light. So you have this photosensitizer, you administer the photosensitizer, then it distributes. Then once it accumulates in your tumor, you irradiate it, and then your cancer cells are dead. Okay. So immunotherapy and photodynamic therapy are two emerging modalities. Hunt for cancer cure. So as I told you, I'm sure you must have realized that cancer is a multidisciplinary problem. Okay, it is not something that can be solved by people or scientists from one discipline. As I have mentioned, chemists, pharmacy wale nahi hai, synthetic chemistry wale, chemists, biologists, pharmacologists and toxicologists and doctors, oncologists, surgeons, radiation doctors, all of them have to work together. And I am sure that if all these disciplines work together, we will have a cure for cancer, just as we have been able to eradicate diseases like polio, COVID. So there is hope. I would like to acknowledge my group members, my advisor, Dr. Malay Patra. I have a very good, we have a good, mix of chemists and biologists in our group. So I would like to thank all of them. I would like to thank my Department of Chemical Sciences, our collaborators, and of course, SERB India, TIFR, and Department of Atomic Energy for the generous funding. Thank you very much for your kind attention.
The podium is now open for questions. Yeah.
Namaskar, sir.
Namaskar.
Sir, while searching for some,
Where?
सर मार्केटमध्ये हल्ली ते नॅनो टेक्नॉलॉजी आणि लायपोसोमल डिलिव्हरी असं मी बघितले. हो, तर या संदर्भात पण काय चालू आहे का याच्यामध्ये?
कसं आहे की तुम्ही म्हणाल्याप्रमाणे नॅनोथेरपी किंवा नॅनो मेडिसिन जो आहे तो अतिशय विकसित, अतिशय रॅपिड रेटने विकसित होणारा विषय आहे. पण तुम्ही जर FDA अप्रूव्ड हे जे मॉलिक्युल्स आहेत जे मी दाखवले, हे FDA अप्रूव्ड ड्रग्स आहेत. ओके, आणि ड्रग डेव्हलपमेंट प्रोसेस जी आहे ती फार लांब लचक प्रोसेस आहे. त्यामुळे नॅनो मेडिसिन मध्ये फक्त एकच FDA अप्रूव्ड ड्रग आहे, अब्रॅक्सेन म्हणून त्याचं नाव आहे. त्यामुळे नॅनो मेडिसिन मध्ये रिसर्च अजून करणं गरजेचं आहे.
थँक्यू सर.
Sir, yeah.
Where?
Hi.
Yeah.
We just went through COVID phase few years ago, and
Huh.
seems the vaccines now have
Right,
been made
right.
to what we learned was targeting specific proteins on the
Yes,
cell.
yes.
So, is there anything like that for cancer cells where
You mean to say, is there anything like a vaccine for cancer?
I don't know how to call it, but it's just targeting specific. What we learned from the virus was basically it was targeting the
Yes.
boundary proteins,
Yes.
and that's how it was to.
There are
different-
targeted therapeutics. So there are targeted therapeutics. So there are certain proteins that are upregulated on cancers, okay? Or they are like specific to cancer. So there is an area that is called as antibody drug conjugates. So that exploits certain proteins that are only expressed on cancer cells and they are not on normal cells. They are not expressed on normal cells. So if you can leverage these specifically expressed proteins, you can have. Yes, they are different, and that makes cancer even more complex. It's just the heterogeneity. From person to person, there is a variation, and even within a person, within a tumor, also you have heterogeneity. Yes, that is where the world is going. You are absolutely right. You're talking about personalized medicine. That is where we are going, towards personalized medicine. And in fact, if you look at immunotherapy that I talked about, immunotherapy, it utilizes your own immune cells. That's kind of personalized medicine. So yeah, we are going towards personalized.
Excuse me.
Yeah. Hello.
Where? Huh.
Ferrocin is a molecule which actually has been introduced as an anti-cancer to replace the platinum-based drugs because of the high level of toxicity.
Yes.
But in your hybrid molecule, you combine both ferrocin and platinum. How does it logically fit into?
How does it logically fit into? Okay. See, it is not, first of all, ferrocin as such. It is a derivatized ferrocin. And we have the toxicology study, these things, body weight and survival things. So from the toxicology studies, we are observing that this hybrid is not toxic to the mice. Yes. No, toxicity comes down. See, toxicity is something that is dependent on dose. And if you have a hybrid material, so let's say this kind of molecule, this hybrid, if you are able to get the same anti-cancer effect at a lower dose, then you could mitigate the toxicity issues.
So does your study show that the platinum dose comes down by using ferrocin?
We are currently working on that.
Okay.
Yeah.
What study apart from body weight you have done to assess the toxicity?
Okay. So body weight is one thing, and then what we do is histopathological analysis.
I beg your pardon?
Histopathological analysis.
Okay.
So we take the cells, we do a histopathology analysis in the compound treated group, and then we observe for markers which could refer to inflammation or damage or something. And looking at the levels of those markers, we arrive at a conclusion whether you have toxic side effects or not.
Instead, can there be any drugs which can overcome the toxicity of platinum-based drugs? Because there are already drugs which have been introduced, like hybrid therapies which have been introduced like that.
Right. So you could go to non-platinum-based molecules.
No, ferrocin is one exactly like that.
So I think in the presentation, I showed these ones. So we have iridium-based molecules, rhenium-based molecules, which can overcome or which can circumvent the platinum-based toxic side effects. Because ultimately, what you need to ensure is that the molecules that you have, these molecules, they go and hit some other axis in the cancer cell. Apart from that.
That's exactly the reason why I'm asking. When you're using a hybrid,
Right.
as a concept, why choose platinum? Why not something else with ferrocin? What is the logic for that? That's the reason I
You
asked.
can see
Because
that
there are
we
so
have
many
also
options
worked-
available.
We have also worked on ruthenium ferrocin.
Okay. Okay.
Yeah.
Thank you.
Yeah.
Hello.
Okay, I
Here.
think we can have just one more question.
Hello. Here. Here. So I saw your presentation. I see there are some gaps in your work, as in you have not covered monoclonal antibodies, and there are many other things, as in not all cancers are same. The whole body, every organ can get affected, and
No, I did not get your question.
so
Could you
it's
kindly
mainly
repeat?
related to MABs, monoclonal antibodies,
Okay.
and yeah, that is the observation. And could you please comment on how Yuvraj Singh got the cancer mediastinal, but his cancer was actually seminoma. So I
Okay.
mean, I have also
Huh.
studied in the same manner. That's why
Huh.
I'm asking.
Okay. No,
It's a
at this
strange-
point, I may not be able to comment on the exact nature of the cancer because.
It's a strange thing. Seminoma, that too in the mediastinum, in the lungs.
Okay. Huh.
He got the
Yes.
tumor in the lungs.
Yes.
So we'll maybe because
No.
bleomycin and
Bleomycin is a molecule that is typically used for this lung carcinomas. Yeah. But yeah, I mean, I may not be in a position to give you more details on this. Maybe we could have a discussion later.
In vivo picture, you talked about how you harvest tumors in mice.
Yes.
So how do you exactly harvest tumors?
How do we exactly harvest tumors? So as I mentioned in my presentation, we first give the subcutaneous injection. We allow the tumor to grow, and then once we monitor the dimensions of the tumor by using vernier calipers. Okay, so that's how we track the growth of the tumor, and then at the end of the study, we sacrifice the mice. Yeah. Sacrifice.
Okay, I think rest of the questions you can ask through the outreach mail that has been provided, because we are running short of time. We have to invite the next speaker. So once